Hi everyone, my name is Fairweather. I'm a PhD candidate with Nigel, Raine, and Jonathan Schmidt in the SES department, and I'm here to talk to you today about ants in agriculture, why I care, and how I get others to care as well. This presentation is going to be broken down into two parts. I'm going to first talk about my research and what I'm doing with the SES department, and then I'm going to breach into my public outreach and how I get involved in the media. Getting into my research, we first have to start at the topic of biodiversity and agriculture. So biodiversity is tightly linked with the function of an ecosystem. Typically, as you get more biodiversity, you get more productivity out of a system or more natural resources. In agriculture, it's not always possible to maintain biodiversity, though. There are some agricultural pests that we have to keep and control. What are the consequences of pest management on the beneficial insects, though? What are the consequences of trying to do this on those that we actually want to keep around? And my study organism within that narrative is ants, Hymenoptera Formicidae. Ants are leaf litter, soil, and vegetation-dwelling arthropods. They occupy a diverse range of trophic levels, from generalists to predators. There are even some social parasites within the group. They're important for agriculture for a number of reasons. They're invertebrate pest predators, so they feed on things like caterpillars and cucumber beetles and aphids. They're soil aerators and nutrient cyclers. They make a lot of tunnels and mounds, and that moves a lot of soil. They're decomposers. They bring things from the surface into their nests, organic matter. They are also pests, though, so they tend to mimic aphilic pests like aphids, and that tending behavior increases their abundance on crops, which could be a detriment to the health of the plant. My project specifically has three main goals. I want to look at the biodiversity and ecology of ants in these agricultural systems. I want to identify the potential routes of exposure of ants to pesticides, and then to test how these pesticides impact ants in an acute and sublethal chronic level. My research so far has addressed the first question, so what is the biodiversity and ecology, and most of my talk is going to focus on that. I have four main field sites for this research within Ontario. There are Strom's Farm and Bakery, Boetcher Farm, Spring Ridge Farm, and a field research site in Lakefield, Ontario. When I get out to my field site, I use a standard method for the collection of leaf litter and soil-dwelling arthropods. I lay out a 50-meter transect. Every 10 meters along that transect, I lay out pitfall traps. I lay out bait traps along those transect every 10 meters as well, of tuna and cookies. I do an hour of active collecting. And then I do leaf litter sifting and soil sampling as well. All the while, I'm paying attention to the ants, what their behavior is, what they're doing, where they're found, and making note of that. Based on these collections in this field research, I've sampled 590 specimens from 17 different species, and we found that the most abundant species on these agricultural crops was Lasius neoniger and Perennialis imparis, two big species. These species were found so abundantly that I started to pay attention to how many plants were occupied by these ants, and Lasius neoniger in particular was found nesting in the majority of the plants along my transect, and this became increasingly true as the season went on, and in fact, in Strom's Farm and Bakery, almost every plant was occupied by an ant nest by the end of the season. So really tightly linked to these crops. So we found Perennialis imparis and Lasius neoniger most abundant. Ants in general were really abundant and present on these agricultural fields. On the bottom there, we have a video of Perennialis imparis on a bait trap in the middle of a 5-acre field. So really, they're really abundant around these fields just in general. I found them doing a variety of things. So they were feeding on pollen and nectar from the plants. They were doing leaf patrols to find pest species on the crops. This was to feed on things like caterpillars, cucumber beetles, and aphids. They also were nesting tightly with the root systems of the plants. So At the base of every one of the plants, we found little tunnels, which the ants were using to bring some of those pest species in and that kind of thing. So we found Lasius neoniger super abundant on these plants, and I decided to take them into the lab and use them as my model organism for the ants in these systems for the subsequent ecotoxicology research. Right now, I have 27 colonies. I started to do laboratory control trials, seeing if they would survive our synthetic soils, our synthetic diet, and we had really huge success. There was less than 1% mortality in the colonies due to these changing conditions. And then the next step was to look at testing these colonies with pesticides, namely neonicotinoids and rhinoid pesticides, and see how the ant behavior changed. But COVID said, not so fast. And so I have all 27 of my colonies at home with me right now in my little mock living room laboratory. And that's kind of the bridge into the second part of my talk, which is how to connect with media. And over the past few years, I've been able to connect with them really effectively, I think. And I'm going to use this situation with COVID and what's been going on as a good case study for how I've gone about and done that. So right now I have ants in my home. I have 20,000 ants. And anytime I share about this, whether it's to news outlets or the media, I always get the question, how do you manage that? How is your landlord dealing with that? How is your husband dealing with that? And that's a very human question, right? It's your experience, not just your science. And so I want to get across the point that you do science, and it's great to talk about it objectively and critically, but you're also a human and you have an experience, and you are you have feelings about your research and what's going on, and you're excited, right? You're associated with your project for 2 to 4+ years. In that time, your passion should come through, and you should be able to share about that in a really experiential way. And I think the media and the public pick up on that quite a bit. So make your science relatable. And it's honestly really easy to connect with the media. If you have something exciting that happened, like I say, like maybe you had something unique happen in the lab a particular day, COVID is a great time. How has your life changed because of COVID? Or you have a paper published or coming out and you want to share about that, reach out to them. They have contact information on their websites. Just send them an email, and they're more often than not open to that. The University of Guelph is a really great example of how to get your research out there. If you just connect with the Guelph media accounts on social media or just send them an email, they are always happy to post about it on their website. And that can often get you more connections. And that kind of breaches into social media. So social media is a great way to connect with media in terms of news outlets and stuff, but also just is a great public face. Facebook alone has 2 billion users on it, and that's a lot of eyes that can be on your research. We're going to have a great keynote talk by Morgan Jackson, and he's going to talk a lot about how to use social media, so I'm not going to get too into it, but I wanted to give a case example of what's happened to me recently and how I've gotten my research into the news because of that. So I posted about some research I was doing with a Harvard professor on Twitter in a thread, just saying the Harvard professor couldn't come to Canada because of COVID. And so I was doing part of the research, and this is how we're coping as scientists. And I tagged at the bottom of the thread a whole bunch of CBC interviewers, CBC accounts, big science influencers on Twitter, and the University of Guelph. And because of those connections that I made and I put it in front of people's eyes, I got an interview with As It Happens on CBC Radio, which has 100,000 plus viewers on their Twitter account. So it's a good example of a way to get your research out there, right? Is just sharing your experience as it happens and connecting with people. And part of that, and I think that's my dialogue associated with all of this, is to connect to yourself personally, if you're comfortable with that. If people know you have hobbies, or you are of a certain identity, or you're part of a certain culture, and you want to connect that with your research, say through writing, dancing. There's a great poet in our department, Madur. There's a lot of different ways you can connect your research and your hobbies, and then put that out there on platforms like social media, and it's going to be broadly accepted and really well received. This is a example from myself. I connect my research with my hobbying, or my costuming hobby, and it's gotten a lot of great reception. I just gave a talk recently with Dr. Wildlife, another researcher from the US that does this as well, at a science conference, and it went over really well. So I encourage you to connect yourself more personally and share that enthusiasm and that human connection with your research. It'll do you wonders with getting eyes on your research. I want to thank the Raine lab and the Schmidt lab for all their help with my project. I want to thank all my funding sources in the University of Guelph for being able to do my research. And if any of you have any questions, feel free to reach out to me on my email, as is on the screen, or connect with me on social media. I'm always happy to talk more about this kind of material and my research. Have a great day. See ya.